Hello, everyone. Welcome to the second part of the video series on coronavirus. Now, in the first part of the video series, we have, tried to define what are microorganisms. Then we have also learned about virus, how to define a virus, then what is the genetic component of virus, and then how a virus is going to cause infection in a host cell. So this much we have discussed about virus. And then we have also discussed about the defense mechanism of a human being, and which is the immune system. Then we have also learned that immune system is not only made up of cells, it is also made up of organs and various other proteins. And all these cells, organs, and proteins, they're going to work together in order to prevent the spread of infection inside the body, or they try to kill the pathogen once they enter inside the system. So this much of the things that we have already discussed in the first part of the video. In the second part, today we will discuss entirely on coronavirus. What are coronavirus? How many coronavirus has been identified till now? What kind of diseases coronavirus they usually cause in human beings? What is the mode of infection? And what are the steps we should take in order to stop the spread of infection? So these are the important areas we are going to discuss in the part 2 of this video series. So let's start. Excuse me. Now, coronavirus is a big family of different viruses. So they belong to the family which is called as Coronaviridae. Now, some of these coronavirus, they cause common cold in humans, and they are also responsible for causing diseases in cattle, birds, bats, horses, etc. Now, till now, there has been 40 coronavirus that has been identified, out of which at least 7 are known to cause infection in humans. The first coronavirus that was discovered was in 1930s in domesticated chicken, in which they were causing acute respiratory disease. The first coronavirus discovered in humans was in 1960s in the nose of people suffering from common cold. So from these 2 points, at least you can make out that coronavirus usually causes respiratory disease in both animals and in your humans. Now, the respiratory tract infection that they cause in human, the magnitude of which can be mild, or it can also be lethal, like the one which was caused by SARS in early 2000, or the one which is prevalent now, right, which is called as the COVID-19. So the magnitude can range. It can be mild also, which may not require so much of attention, or it can be lethal also. Sometimes it can be life-threatening, and sometimes the person needs a lot of medications in order to become well. Well, we have these many coronavirus that causes common cold in humans. That includes 229E. Then we have OC43, SARS-CoV, HCoV, NL63, HCoV, HKU1, MERS-CoV, and SARS-CoV-2. Now you have to understand that these are different strains of coronavirus. So we have coronavirus, and as I have mentioned in the last slide, at least 40 coronavirus has been identified till now. So what are these 40 coronavirus? These are different strains of the same virus. So these are the different strains, okay, which is going to cause common cold in human. All these coronavirus is zoonotic, which means coronavirus, they usually spread from animals to humans. So that is the meaning of zoonotic. So when it is spread from animal to human, then we use a term which is called as zoonotic. Now let us learn about the genome of the coronavirus. Now in this particular slide, we have the diagram of a typical coronavirus. Now coronavirus is an envelope virus having RNA as the genetic material. So coronavirus, we know now it is an RNA virus. The viral particles usually measures approximately 120 nanometer in diameter. Now, why this particular name was given? Why coronavirus? The name was given because of the crown-like projections that they have on the envelope. Now, in the last part of the video, I have already mentioned that these are nothing but these are the glycoproteins which are expressed on the surface of the envelope. And what is the importance of glycoproteins? These glycoproteins are going to help in the attachment of the virus to a host cell, which is very important. Okay, so remember, we have learned about different steps of viral infection. The first step was attachment. Now, how they're going to attach to a particular host cell? With the help of these glycoproteins, which are expressed on the envelope. Now, because of this unique, crown-like appearance, okay, because of the unique crown-like appearance of these glycoproteins, the name coronavirus was given. So in Latin, the name corona means halo or crown. So because of this crown-like appearance, which are nothing but these are glycoproteins only, which is going to help the coronavirus to attach to host cell. So the name coronavirus was given. What is the genome size of this virus? Then the genome size approximately ranges from 26 to 32 kilobases. Now we have to know that 1 kilobase is 1000 base pairs. Imagine how big is the genome of this virus. Okay, so it ranges from 26 to 32 kilobases. Okay, so again 1 kilobase is equal to 1000 base pairs. Now, the envelope of the coronavirus is made up of lipids, which means it is easy to kill a virus simply by washing our hands with alcohol, or we're going to wash our hands with soap. Okay, because remember when we have oil on our hand, how to remove the oil from our hand, we use soap. Now here also the coronavirus, the envelope contains what? The envelope of the coronavirus contains liquid. So how to get rid of that? So we can kill coronavirus when we very frequently wash our hands with soap, or we are going to sanitize our hands using sanitizer that contains alcohol in it. Anyway, so inside this coronavirus, inside this envelope, again, we have the capsid, which we have already discussed in the first part of the video. The capsid is made up of what? Capsid is basically made up of proteins. But there is a simple difference in it. In case of coronavirus, the capsid is found in association with the RNA molecule in a continuous conformation. So capsid is obviously made up of protein, that is true. Plus it is, directly associated with the RNA molecule in a continuous conformation. Can you see this? So this is a nucleocapsid, which also is associated with the RNA molecule in a continuous conformation. Now, the RNA molecule that this virus has is a positive sense single-stranded RNA. Now, what is the meaning of this? So when a virus is going to have a positive sense single-stranded RNA, it means that this particular RNA can be directly translated into viral proteins. And these viral proteins are going to help in the formation of viral particles containing the viral genome. And these viral particles are going to cause lysis of the cell, then it is released into the surrounding tissue. Did you understand? So, some of the RNA virus will have positive sense single-stranded RNA. Some of the RNA virus is going to have negative sense single-stranded RNA. Excuse me. So if in case a virus has a negative sense single-stranded RNA, in such cases, the negative sense has to be transcribed into a positive sense RNA. Then this positive sense RNA is going to be translated into viral proteins. Then these viral proteins again is going to give rise to viral particles containing the genome of the virus. Then these viral particles are going to cause lysis of the cell. Then it is released into the surrounding medium. But I think you have to remember that first point is what? The coronavirus is a RNA virus, which means it has RNA as a genetic material. First point. Second point, that it is an envelope virus. And this particular envelope, on their surface, they have the glycoproteins. And because of the crown-like appearance of these glycoproteins, the name coronavirus was given. And what is the what is the RNA molecule that the coronavirus they have? It is a positive sense single-stranded RNA, which means this particular RNA will be directly translated into viral proteins, and these viral proteins will be again forming viral particles. We know the rest of it, right? Viral particles, once they are formed, so they are going to cause lysis of the cell, then they are released into the surrounding tissues, which means they can spread infection. Now, this RNA molecule that the coronavirus has a typical modification. Okay, so this RNA molecule comes with a modification. Now, what is the modification? Now, we all know whether it is a DNA or RNA, they're going to have two ends. One is called as the 5' end because of the presence of the phosphate. Another one is called as a 3' end because of the presence of a hydroxyl group. Is this okay? So this RNA molecule is no different. This RNA molecule is also going to have a 5' end and a 3' end, but these ends are modified in case of coronavirus RNA molecule. Now, what is the modification? In the 5' there is a capping. In the 3', lot of adenine residues are added, okay, forming a tail. So it is called as poly-A tail. Poly means many, right? So many adenine residues are added at the 3' side, forming a tail. So that is called as the poly-A tail. So you have a poly-A tail in the 3' side, and you have a capping at the 5' side of the RNA molecule of coronavirus. Now, why these modifications are important? And these modifications are important so as to protect this RNA virus from nucleus enzyme digestion. Now, from where this nucleus enzyme is going to come then? Nucleus enzyme is usually present in the cytoplasm of the host cell. So when the RNA molecule is going to enter, the nucleus enzyme is going to cause degradation of the RNA molecule. That means it has the capability of degrading the RNA molecule, which means it is going to stop the stop the spread of infection. Now, since this RNA molecule of coronavirus has these modifications like capping at the 5' side and poly-A tail at the 3' side, so this is going to give protection to the viral RNA against nucleic acid digestion once it enters inside the host cell. All right. Now, the genome of coronavirus is going to code for proteins and enzymes. All right. So certain enzymes like replicase, transcriptase, certain structural proteins, they're going to code for, for spikes. Spikes are nothing but the glycoproteins, which is expressed on the surface of the envelope. Then membrane proteins, nucleocapsids. So all these proteins and enzymes are coming from the genome of the coronavirus, which is the RNA molecule. Again, you have to remember it is a positive sense single-stranded RNA, plus they have modifications also. 5' side they have a modification, and the 3' side also they have a modification. All right, so this is a a structure of a coronavirus. Now this is a pictorial representation of how the virus is going to enter, where exactly they cause infection, how they spread infection. Okay. So as we all know, the coronavirus is going to enter through our mouth or through our nostrils. It is not going to enter through our skin. Okay. So coronavirus basically enters through our nose or mouth. So it is going to go inside, and this is the respiratory tract. So in the respiratory tract, we have a membrane, which is called as a mucous membrane. Now, what is the function of mucous membrane? Mucous membrane is basically going to release a sticky substance, which is called as mucus. The importance of mucus is what? It is going to wash away any sort of pathogen that has entered through our mouth or through our nostrils. But in this case, it is difficult for the mucus, to wash away the virus. So what happens? The virus attaches itself to the membrane. All right, it enters inside the cell. It undergoes replication. Then, because of its infection, there is an inflammation of the respiratory tract, which is seen. And because of which the person who is infected by the virus, they have this breathing problem. I think it is quite common these days. So anyone who is having breathing problem, doctor prescribes that you should get your checkup done because you might be having the virus in you. All right, so this breathing problem is one of the very common and important symptoms that is seen with people who are infected by the virus. Now, why this breathing problem is there? Because there's an inflammation of the respiratory tract through which the air is going to flow. Now, since there is inflammation, the air cannot flow very easily, and because of which the person is going to experience a lot of breathing problem. Anyway, from the respiratory tract, it is going to enter inside a lung, where it is going to spread more infection. Then from the lung, it is going to enter into our blood. So in the blood, again, since blood is flowing in the entire body, then they start spreading infection in the entire system. So this is just a very simple representation of how coronavirus is going to enter and what exactly happens when coronavirus enters into our system. Okay. Now this picture, it defines all the steps of viral infection. Okay, it may look very confusing, but let me explain it. It's not very confusing. Let me explain you. Now remember, I told you like the first step of viral infection is what? It is attachment. So the virus has to attach to a particular host cell. And how the virus is going to attach to a host cell? With the help of the glycoproteins that they have on the surface of the envelope. Okay, so these glycoproteins play a very important role in this viral attachment. Now how are they going to attach to the membrane of a cell? It is going to attach with the help of these receptors. So first of all, the glycoproteins will bind to the respective receptors which is expressed on the surface of a host cell. So this is a host cell membrane, okay, which has the receptors on it. So the glycoproteins are attached to these receptors. So this is the first step. So attachment. After attachment, what is going to happen? The viral part, the virus is going to penetrate, or it is going to insert its genome into the host cell. Now remember, I told you what was the genome of the coronavirus. Genome is RNA molecule, but it is not any RNA molecule. It is a positive sense single-stranded RNA. So here you can see it is written as positive strand. Okay, so this is a positive sense single-stranded RNA. Now the cytoplasm of the host cell will contain organelles which are called as ribosome. The ribosome is also called as protein factory because why? Because these ribosomes are going to help in the formation of proteins. We've been hearing that we have proteins and enzymes and everything. So all enzymes are protein except one. Anyway, so these proteins are formed on these very tiny organelles which are called as ribosomes. So what is the name I said about ribosome? They're also called as protein factory. Anyway, so as soon as the RNA molecule of the coronavirus enters, it is going to take the help of host ribosome in order to produce its first protein, which is an enzyme. And what is this enzyme? It is a polymerase enzyme. Now, why they need a polymerase enzyme? Because they have to, multiply their genome. So what happens? This polymerase enzyme is going to form a negative strand. So from a positive strand, using this viral enzyme is going to form a negative strand. Some of the negative strands are going to give the viral proteins, and some of the negative strands are going to form the positive sense RNA. Did you understand? Now, the proteins which were formed from this negative strands, they are going to help in the formation of the viral particles. And this viral particle will be having the positive sense single-stranded RNA molecule. So once the viral particles are formed, then what is going to happen? What was the next step? Now these viral particles need to be released from the cell. So how they will be released? They will cause lysis of the cell. Then they are released into the surrounding tissue. So in the surrounding tissue, they can start spreading infection. Again, the same process is going to take place. What is the first process? What was the first step? It is going to attach to a nearby healthy cell. Then they're going to insert its genetic material. All right. So again, this whole process starts in a new cell. So that's how coronavirus is going to spread infection inside a host organism. Okay. So you have to remember that this is a positive sense single-stranded RNA, which is directly translated into its enzyme with the help of the ribosome. This ribosome is of the host cell. Okay. And then this polymerase enzyme is going to form a negative strand. Some of the negative strands will give the viral protein. Some of the negative strands will undergo again transcription to form a positive strand RNA molecule. Then all these together, that means your proteins, viral proteins along with the genome is going to form a viral particle. And these viral particles, in order to cause infection, what they have to do? They have to be released by the cell. So they're released by the cell by causing lysis. Then once they're outside the cell, they will bind to the nearby healthy cell and they will again start this whole process. Okay, and that's how they're going to spread infection inside an organism. Now let us discuss what is COVID-19. Now we've been hearing about COVID-19 in news and newspapers. Every day we've been hearing about COVID-19. Now COVID-19 is not a virus. COVID-19 is what? It is a coronavirus. So coronavirus has been abbreviated as COVID-19. All right, so COVID-19 or coronavirus disease is an infectious respiratory disease which is caused by newly discovered coronavirus called SARS-CoV-2. Remember in one of the earlier slide, I have said that there are, whatever coronavirus that causes respiratory infection in humans, all these are different strains of the same virus. Now this is a different strain, okay? So which is related to SARS. Now SARS was causing, again, respiratory disease in early 2000. So COVID-2, that is coronavirus 2, is quite related to SARS. So that's why the name was given as SARS-CoV-2. Anyway, so what is COVID-19 again? COVID-19 is coronavirus disease. What disease it is going to cause? It causes infectious respiratory disease. And who is causing this disease? SARS-CoV-2. Now we all know that this particular virus originated in bats, and people who were consuming these bats, the virus got transferred from the bats to the humans. Remember one particular term we use in one of the slide, which is called as zoonotic. What is the meaning of zoonotic again? So when the virus is getting transferred from animal to human, so that term we use is a zoonotic. So in this case also it was no different. Okay, the virus actually came from bats only. And which city of China was the epicenter? It was Wuhan city. Now we know it has started spreading. It has spread in most of the countries around the globe. So it has now become pandemic. Okay, that's why the term it is used, which is called as infectious. It is highly infectious. All right, it spreads so quickly and so easily that many people has been infected by this particular virus. And what was the name of the virus? SARS-CoV-2. All right, so you have to remember that yes, it originated in bats. Now what is the present scenario? The present scenario is that it is not only transferred from bats to human. Now humans are responsible in transmitting the virus, all right, from an infected individual to a non-infected individual. Now when a person is going to be infected by the virus, all right, there are a group of people who is going to show the symptom. Now what is the symptom? I said number one is breathing problem. Number two, the person is going to have runny nose. That means he's suffering from cold. The person is going to sneeze. The person is going to cough. So he has the infection in his body. Okay, so he is basically showing all the symptoms. So those are called as people who show symptoms. But there will be certain people who are not going to show any symptoms, but they will be carrying the virus in them. Did you understand? So both people are infected, and both the people are equally responsible in transmitting the virus from them to a non-infected individual. Did you understand? So earlier it was known, it was correct that the virus came from bats to humans. Now humans are responsible in transmitting the virus from an infected individual to a non-infected individual. Now if you see a person is normal, he is not showing any kind of symptoms, but you never know, the person might be carrying the virus in his system, so he is also infected. So if the person is going to sneeze and if you stand next to him, so you can also get the virus into your system. So the person is also responsible in transmitting the virus to you. So nowadays it is human which is responsible in spreading the infection worldwide. Anyway, so most people who are infected with this virus experiences mild to moderate respiratory illness. Most people recover without needing any special treatment. Remember in one of the slides I said it causes common cold, but the magnitude of this particular disease can be mild also, all right? Or it can be highly lethal. That means some people, however, become seriously ill and experiences difficulty in breathing. So breathing problem, okay? Difficulty in breathing is one of the common symptom which is seen in people who is infected with SARS-CoV-2. Did you understand? Since it is related to our respiratory system, and I showed you one diagram also, you know, when it starts infecting the respiratory tract, inflammation occurs. Inflammation is going to obstruct the airflow, and that's how the person is going to have breathing problems. So breathing problem is one of the common symptoms which is seen in people who is infected by this virus. Apart from breathing problem, what else is there? Now we have fever. Fever is common, okay? Then dry cough, tiredness, body ache, nasal congestion, sore throat, or diarrhea. Now one thing you have to understand, that dry cough, okay? Dry cough, then we have nasal congestion, sore throat. These are all related to upper respiratory tract. Now our respiratory system is divided into two. One is the upper respiratory tract, another one is the lower respiratory tract. Upper respiratory tract will include your nose, mouth, you know, the respiratory tract, eyes. So these are the upper respiratory tract. When we say lower respiratory tract, that includes the lungs, okay? And lungs has what? Lungs has bronchioles and alveoli. So these are the lower respiratory tract. So one thing we have to understand, that coronavirus causes infection in the upper respiratory tract. So it is always given in the news. So if anyone is experiencing breathing problem, along with breathing problem, what they have is dry cough. Did you understand? Not a wet cough, it is a dry cough, nasal congestion, sore throat. Then he should immediately get himself checked by a doctor. So he might be carrying the virus in his system. Now fever. Now what happens? Fever is nothing but when the body temperature increases. Now fever is one of the defense mechanism of our body against the pathogen. Say suppose a pathogen has entered inside your body. Now these pathogens, they grow very well at the typical temperature, which is our body temperature. Now our body itself takes a step by increasing the body temperature. So when our body temperature is increased, so it is going to make the environment for these pathogens to grow unfavorable. That means these pathogens will, cannot grow at a temperature which is higher than the optimum temperature. Did you understand? So you have to understand that when we have fever in our body, it means that we have an infection in the body. Now how it is related? Because as I said, when there is an infection in the body, our body itself takes a step in order to decrease the spread of infection. How can we do it? By increasing the body temperature. Anyway, so fever will be there in any kind of infection. Apart from it, as I mentioned, coronavirus, that is SARS-CoV-2, is going to cause disease in upper respiratory tract. So dry cough, nasal congestion, and sore throat is very important. Apart from it, what is important is breathing problem. The person should be having difficulty in breathing. So these are certain common symptoms which is seen in people who are infected by this virus. Now you see, it takes so many days for a person to show symptoms. So symptoms may appear between 2 to 14 days after exposure. So sometimes person may be carrying the virus, but he's not showing any symptoms because, as I mentioned, but he is having the virus in his system. Okay, so even that person is responsible in transmitting the virus to a non-infected individual. Did you understand? So he is equally dangerous. Just that he's not showing any symptoms, but he's carrying the virus in the system. Okay, so he's equally dangerous in transmitting the virus to a normal, non-infected individual. Now, what group of people are highly affected? Older people. Then people with underlying cardiovascular disease, a chronic respiratory disease, cancer, and then people having pre-existing medical conditions. Now, what is common in all these people? The immune system is weak. Now, older people, okay. So when you cross a certain age, the immune system becomes weaker and weaker. So people with weak immune system are more, vulnerable against this particular infection. All right, say suppose a person is having some sort of pre-existing medical condition, so he's maybe taking medicines, the immune system has gone weak. So those people are more susceptible in taking the infection, all right? Because since the immune system is weak, now the virus can easily spread, because our immune system, as I mentioned, this is the defense system of our body. So they will try to defend the body against this infection. Now, since our defense system is so weak, the virus can easily enter inside the body, and they can easily start spreading infection. So anyone who is having weaker immune system, especially older people, or people having, you know, cardiovascular disease or cancer, or any sort of pre-existing medical conditions in which the immune system is weaker, all right, so those people are more dangerous, or sorry, not dangerous, and those people are more vulnerable towards that particular disease. The disease is going to spread very easily in such kind of individual, all right? The COVID-19 virus spreads primarily through droplets of saliva or discharges from the nose when infected person is going to sneeze or cough. These tiny droplets, which are difficult to see, travel in the air and lands on nearby person or on surface. And these 2 points is particularly important, because now we've been telling that infected person can, transmit the virus to a non-infected individual. But how that is possible? Now, say suppose a person is infected and he has a virus in his system. Now, those person is going to, remember I told you these viruses, they cause common cold, which means they're going to sneeze or they're going to cough. So when a person is going to sneeze, he's going to discharge lot of, droplets into the air, which cannot be seen at all. So when a person is going to see, have you ever observed any kind of droplets in the air? It is not possible. We cannot see, because these droplets are so very tiny. These droplets are going to carry the virus in it. Now, if you're standing next to that person, then what is going to happen? Now, these droplets are so tiny, it is going to travel in the air. Now, if you're standing next to the person, you are going to inhale air. So along with the air, you're also inhaling these droplets, which is going to have the virus in it, which means what? Your system also has the virus. But sometimes what happens is, suppose you're not standing next to the person, you're standing a little away from the person. The person is going to sneeze. It is going to travel in the air for some time. Then because of gravity, these droplets are going to fall on certain surface, all right, metal surface, rubber surface, plastic surface, any kind of surface. So it is going to travel for some time. And because of gravity, it is going to fall onto the surface. Now this time you were inhaling, but you did not inhale the droplets. But what you did is you have touched the surface with your bare hands. And people, by very nature, they have this habit of rubbing the face, touching the nose or eyes or the mouth. So what is happening? You touch the surface, your hand has the virus in it. Now you are touching the hand. With the same hand, you're touching your nose and your mouth, which means what? The virus is again entering inside your system. So either way, the virus can enter inside the system. So we have to take a lot of precautions. One, what is the first precaution? That we should not go out. If you're going out, then we have to cover our face, and we should not touch our face with our dirty hands, because our hands may contain the virus, because virus cannot be seen. You may have touched certain surface which may have the virus in it. Okay. So this is very important. Okay. So how this particular virus is spreading? Either it is spreading through air, that means it is present in the droplets which is discharged from the nose of the infected person, or else it is going to travel in the air for some time, then it is going to fall on the surface because of gravity. So it can be present on the surface also. When we're going to touch it, and with the same hand we are going to touch our face, it is also going to enter into our system. So either way, it is going to enter inside our system. So it is important that we should take a lot of care. Now, how to detect a person is having a coronavirus, the virus inside the system. Say suppose we have seen some symptoms in the person. Now we need to detect whether the person has, is it because of the virus or it is because of something else? Because common cold maybe because of some other reasons also. Now we have to test whether a person who has all the symptoms of COVID-19 disease, how to detect whether actually the person has the virus or not. So we have two tests in it. So we're going to do two tests in order to find out whether the person is infected by the virus or not. One is a molecular test. Okay. The second one is a serological test. Now, what is molecular test? In molecular test, we first of all take a swab sample. Now, what is swab sample? It's nothing but it is like a earbud. Okay, so we're going to have earbud, and we're going to take a sample either from the nose or from the throat. Because remember, I told you these are the major areas through which the virus is going to enter. So our nose or the throat of the infected person will have the virus in it. So for molecular test, we take a swab sample. Again, what is a swab? It is just a earbud only. So either we're going to take the swab sample from the nose, or we are going to take the swab sample from the throat of the infected person, of the a suspected person. We can say it is a suspected person. We do not know whether the person is infected or not. We have to find out if the person is infected by the virus. Okay, so we can use the term as suspected individual. And after that, what we do is we go for reverse transcriptase polymerase chain reaction. This is a very complex, a technique which I'm not going to explain now. Anyway, so what we do is we take the sample, we go for RT-PCR, and this particular test is going to detect the presence of the viral genetic material. Did you understand? And if they identify two specific SARS-CoV-2 genes, then that means it is a positive case. This is okay. So let us use the term as a suspected individual. So we have suspected that the person might have the virus. In order to find out, what we're going to do first is a molecular test. We take a swab sample, we go for RT-PCR, and this one is going to help in the detection of the presence of the viral genetic material. And if in case they have identified two specific genes of this particular virus, the case is confirmed. What is the confirmation case? That the person is infected by the virus. Okay. What is the second test? Second test is a serological test. And in this case, we are not going to take swab sample. We are going to take blood sample. Serological test means the test related to the blood serum. Okay. So in this case, we are not going to take the swab sample. What else we take then? We take the blood sample and we try to find something in it. What we try to find are the antibodies which has been produced against the virus. Say suppose a person has been infected the virus. That means the virus has entered inside the system. The virus has activated the immune system of the individual. Now, what happens after the activation of the immune system? Antibodies are produced in the system. So serological tests try to find out whether the person has antibodies which are specific against the virus or not. If the person has those antibodies which are specific against the virus, which means it is a positive case. Did you understand? So again, if a person is having mild symptom, then also the antibodies specific for this virus will be present. Or if the person has already recovered, okay, the person was infected, he has taken medicines, he has recovered from the disease, still the person's blood serum will contain the antibodies specific against the virus. Did you understand? So this is a second test which can be used in order to find out whether a suspected individual is having the virus or not. What is it again? It is to find out the presence of antibodies which are produced against the virus. Once the virus is entered, as I mentioned again, what happens to our immune system? It gets activated, right? Once it gets activated, they will produce a lot of antibodies. So these antibodies are specific against the virus. So antibodies, all antibodies are specific, okay? Some other virus has entered, this antibody won't be there. Some other antibodies, specific antibodies will be present, specific for that antigen. But in this case, if the virus has entered, antibodies will be produced against a specific virus, the presence of which is going to tell us whether it is a positive case or a negative case. Now, as I said, some people may have mild symptoms. He will have antibodies in his system. The person has recovered also, still he is going to have antibodies in his system. Anyway, so these are the two tests by which we can find out whether a suspected case is a positive case or it is a negative case. Now, how can it be prevented? Now, this is important. Okay. Now, we've been discussing about coronavirus. It spreads in the air. Nowadays, human beings are responsible in transmitting the virus from one that is infected individual to non-infected individual, and that is basically causing the widespread infection. Okay. So many people has been infected by this virus. Now, how that is possible? Because number one, population is high. We are not taking extra care. Infected people are roaming around, because I told you some people may have the virus, but they're not showing any symptom. They may not know that they are carrying the virus. They're sneezing, they're coughing. The people who is standing next to him, he's not taking any care, so he's inhaling the air. So that's how the particular virus is getting spread in the entire area, in the entire world. Okay, so let us take some steps in order to stop the spread of infection. So these are the preventive measures we should take. The first is washing hands. Remember, I told you soap is going to kill the virus. Now, how soap is going to kill the virus? Because remember, I told you the envelope is made up of lipid. Now, how to remove lipids? We have to use soap. So when we're going to wash our hands with soap, at least for 20 seconds, that is going to kill the virus which has got stuck to our hands. Did you understand? This is the first step. So we have to keep on very frequently wash our hands with soap, at least for 20 seconds. All right. What is the next preventive measure we should take? Avoid touching the face, eyes, nose, or mouth when the hands are dirty. Remember, I told you, if you're not taking any protection, you're not covering your face, you're touching the surface containing virus with your bare hands, then again you touching your nose and your mouth. So that should be prevented. We should not do it at all. We should not do it at any cost. What is the third one? Staying away from people at least 6 feet or more. Remember, I told you, because it travels. First of all, it is going to travel in the air. Then due to gravity, it is going to fall. Now, if you're standing next to the person who is sneezing and you're not covering your face, what is going to happen? You inhale air. Along with the air, the droplets are going to enter inside the system, which is carrying the virus. What is the fourth preventive measure we should take? Covering the mouth with a tissue or inside of your elbow whenever sneezing. So person who is infected, all right, so you have to cover, you just cannot start sneezing in front of someone. You have to cover your, that is basic etiquette, am I right? You have to cover your nose with a handkerchief or maybe a tissue, to stop the spread of infection. That is a basic etiquette we should always follow, not only in this case, every time. Anyway, so this is one of the preventive measures. What is the last one? Clean any objects you touch a lot. Use disinfectants on objects like phones, computers, utensils, dishwashers, doorknobs, and nowadays it is quite famous, hand sanitizers. Okay, now hand sanitizer, these are alcohol based, so whether you use soap or alcohol, they're going to kill the virus. So you should always keep hand sanitizer which are made up of alcohol very handy, and you should keep on cleaning your hand, the surface that you're going to touch, your phone, anything that you are going to touch with this particular sanitizer, which is going to make sure that the surface has been cleaned of the virus. So soap and alcohols are very important. Apart from it, we have to take steps in our body itself, that we should always cover our mouth. We should not touch our mouth with our dirty hands. So these are certain preventive measures. This is a diagram which basically tells you, this is an envelope which is made of a protein. These are the soap molecules, all right? The soap molecules are going to destroy, see, lysis of the viral particles, the coronavirus, and this virus has been killed. So soap is very important. So very frequently we have to keep on washing our hands with soap. You go outside, you carry sanitizer. While you come inside, you wash yourself with soap, or at least you keep on washing your hand with soap. So that is going to make sure that we are getting rid of the virus from our body, and that's how we can prevent the spread of infection. Anyway, thank you so much for listening to this video. I hope everyone is safe. We have to say, stay safe. And how to stay safe, we have to take a lot of precautions. Okay, if possible, try to stay indoor and go out only when it is required. And if you're going out, you have to cover your face with a handkerchief or maybe a mask. You should always carry a sanitizer in your hand. And when you come back to your home, what you have to do, you have to wash your clothes, you have to wash yourself completely, properly, so that you are removing all the virus from your body. All right, so that's how we can stay safe. Thank you so much. In the third video which is coming up, we will discuss about what are the vaccine concepts that has been developed for coronavirus, and what is plasma therapy, and etc. and etc. We will discuss it in the third part of the video. Thank you so much.